Humans have 46 chromosomes in their cells. Those 46 chromosomes actually come in two sets. One of those sets came from mom, and the other one came from dad. Together, this makes a diploid cell. Now, we can see these chromosomes on a picture called a karyotype, which we have here in front. And we can see that our chromosome sets are paired. So this is chromosome number 5, and chromosome number 5 has two chromosomes. One of those came originally from the mother, from the egg, and one came from the sperm of the father. This is the appropriate amount of DNA for the cell to be able to sort through and to use to do regular cell function. However, this is too much DNA to be an egg or sperm. In sexual reproduction, our goal is to make a new baby, or a zygote. That's what we call this new cell that's going to become a baby. In order to do this, we need to merge an egg and a sperm together, and we have to end up with that 46 chromosomes that is a normal cell. So it wouldn't work if the egg had 46 chromosomes and the sperm had 46 chromosomes, because together they will not make 46. Instead, they're going to make 92. That's way too much DNA, and that cell will not survive. When fertilization happens, we need to make sure that when the egg and sperm fuse together, we still get 46 chromosomes. So to get the 46 chromosomes, we need to make sure that we are getting 23 chromosomes in the egg that comes from mom and 23 chromosomes in the egg that comes from dad. Meiosis is a special cell division that creates gametes, egg or sperm, with only one set of data. One set of data is called a haploid cell. And together, after fertilization, there will be two sets in the zygote, which is the baby cell, and it will be a diploid normal body cell. It is a cell division, so how does this cell division actually happen? The process of meiosis is actually 8 steps, but there are only a few that we need to discuss to understand what their relationship is to our traits. We need to see what the cell looks like at the beginning. We need to understand how it lines up and what it looks like after it's split. We're also going to need to look at where it lines up again and what it looks like at the end. So we're going to use a simpler diagram to help us to see this. You have the same diagram on your notes, and what we're going to start with is the cell up at the top. This is the mother cell, the original cell that's either in the ovary or the testes. And in this cell, we have 4 chromosomes, 2 large chromosomes and 2 small chromosomes. Now, in reality, humans have 46 chromosomes, but in this case, we're going to work with 4 to make it a little easier for us to keep track of. Now you can see that the 2 big chromosomes go together because of their size. Both of these chromosomes carry the same type of DNA, so we call them homologous chromosomes. On the red chromosome, there is a gene that says that you're going to have dimples. On the blue chromosome, there is a gene that says you're not going to have dimples. Both of them have to do with dimples. The 2 small chromosomes, the yellow and the green, also carry similar information. The yellow carries information for freckles, and the green carries information for no freckles. The tricky thing is that this person can only pass on 1 piece of DNA for dimples and 1 piece of DNA for freckles. It can't pass on any more than that because that would be too much. So we need to separate all this information away from one another. So in meiosis, the spindle fibers are going to form and attach to the chromosomes, and they're going to begin lining them up so dimples are next to dimples and freckles are next to freckles. And you can see that they're lined up in the middle, but that they're lined up in pairs. And when they separate away from each other, we will be pulling dimples to either side and freckles to either side. So take just a moment and do your own drawing on your diagram so that it's easy for you to remember how these line up. So now we have 2 new cells. Each cell has information about dimples and information about freckles. However, if you look at the red chromosome has 2 copies. That's still 1 copy too many. So we need to go through division again. The spindle fibers form and they pull these chromosomes to the center. Once they're in the middle, the copies are going to pull apart from one another. So stop the video and fill in your notes. When the spindle fibers finish pulling and the cells separate from one another, we can see that we have 4 cells. Each cell has only 1 gene for dimples, 1 gene for freckles. If this is happening in a male, all 4 of these cells will become sperm cells. If this is happening in a female, these cells become eggs. Remember that in fertilization, the egg and the sperm join together. The egg has a gene for freckles and a gene for dimples. The sperm has a gene for freckles and a gene for dimples, and they are going to join together at fertilization. The new cell is going to be diploid, having 2 sets, 1 set having come from mom and the other set having come from dad. This is the basis for genetic prediction. If we can predict the combination in the egg, we can also predict the combination in the sperm. And if we know what those combinations are, we can also predict the possible outcomes in their children.